So no, we haven't talked about the cell structure yet of a bacterial cell, but the significance to the reason why we're talking about biochemistry is to understand, the cell structure of microorganisms. And with bacteria, they have a very specialized structure, or at least some have this structure, some have a very thin layer of this structure. But the structure is known as peptidoglycan. And peptidoglycan is the formation of peptides, which are very small proteins, as well as glycan, which is essentially a carbohydrate that are joined together. And the bacterial cell is surrounded by what's called a bacterial cell wall. And that cell wall is fortified or built using peptidoglycan. Now, some bacteria have this, some bacteria have a lot of this. They might have 40 layers of peptidoglycan, if not more. And then some may have just maybe one layer of peptidoglycan, or 0 to 1 layer. So they may not have peptidoglycan, period. And so this particular structure helps us to distinguish between what's referred to as a gram-negative bacteria and a gram-positive bacteria. So we won't hold off on that for just a second, but the structure itself is important for us to understand because of the chemical attributes that it is made out of. And so here you can see kind of the molecular structure of essentially your carbohydrate molecule, which is the beta portion of the peptidoglycan structure. And then you have kind of this group of what's referred to as kind of amino acids that hold together these different chains. Okay, so alanine, lysine, D-alanine, they link this disaccharide to this disaccharide, like we've talked about. And through that linking, you can have, if you have multiple of these structures all joined together, you end up with a very large, thick layered chain. So each of these chains are just held together, and they surround the bacterial cell, which provides structure for the cell, and it also provides protection for the bacterial cell. And allows us also to identify what type of bacteria we're working with. So peptidoglycan is nothing more than a repeating disaccharide that is attached to proteins. In this instance, we also have nucleic acids that make up the protein. And they form this structure that will surround the bacterial cell, providing protection to it. Okay, so that particular disaccharide is very, very critical to the microbiology realm because, again, it helps us to figure out how to treat certain bacteria and helps us to understand what type of bacteria we are working with. And we'll cover more about differences when we talk about our next unit, our Chapter 3 unit. So polysaccharides are similar to disaccharides in the sense that they're just many carbohydrates joined together. So a polysaccharide is defined by your textbook is one where it's just composed of many monosaccharides, and you can have up to hundreds of these monosaccharides joined together. So two key examples are that of starch and glycogen, which are very large, complex polysaccharide molecules. And polysaccharides are important to an organism's system because they enable a location where energy can be stored. So you basically have these large blocks of polysaccharides that are just sitting around in this organism's system, and our own body. They sit around in tissues and help to build fat deposits in the body. But when the body goes into starvation mode and isn't able to acquire the energy sources it needs, it will have a storage supply. And so in our cells, we have that storage supply is known as glycogen, and in the plant cells, that storage supply is known as starch. So these molecules not only provides the function of being able to provide energy for the organism, but they can also be used to build structures as well as to provide protection. One example of protection is that of bacterial capsules. A capsule is a structure that will surround a bacterial cell, and it will protect the bacteria from being digested or phagocytized by specialized immunity cells in your body. So you have these cells in your body that will just go around eating viruses, eating bacteria. We call them macrophages. But if there's a structure that protects the bacteria from being ingested, that can be used to prevent that from happening, the bacteria develop what's called a capsule that the immunity cell is not able to penetrate or digest. So polysaccharides are very important. They have a number of functions to store energy. They can also provide protection and support to the organism that's using them. Another kind of idea behind polysaccharides is the fact that they are examples of polymers, which are basically just, it's just a molecule or molecular structure that contains very many subunits, and all these subunits are pretty much exactly the same. So you're looking at just kind of a chain of the same monosaccharide molecule that comes together to produce a polysaccharide polymer. Now, just like we talked about with disaccharides, when you have certain enzymes or when you have water molecules being present in the system in which your polysaccharide is, those things can act on the polysaccharide and they will start to break them down. So in this image here, you can see that first molecule is out of a starch molecule. So starch is a polysaccharide, but the bonds between them that hold together the units of the starch can be broken using both water and an enzyme. An enzyme for starch is a specialized enzyme that's called amylase. So the salivary amylase that you have in your saliva is used to help break down starch or any foods that contain starch that you put in your mouth. So something like a French fry, which is just straight from a potato. There's nothing in that except for a whole bunch of starches. Well, the enzymes mixed in with the water from your mouth all create a great environment to break apart those starch molecules, so that way the starch components can be broken down and then be entered into your cells as they go through the process of digestion. And the same thing can happen to a lot of other large polysaccharides like the glycogen that we were talking about. This figure shows you up here what the polysaccharide starch would look like, and then it goes through the process of breaking it apart into smaller molecules. So with this polysaccharide, it's been broken. One of the bonds has been broken. They refer to these oxygen bound to carbon bound to these two monosaccharides as a glycosidic bond. And when water and enzyme hit it, it will split the molecule into two disaccharides. And so now you go from having starch to two molecules of maltose. And then another enzyme comes in and it starts breaking apart the glycosidic bonds between these disaccharides. And then that enables the formation of 4-glucose products. So, and then from here, you can undergo the process of glycolysis, where each of these glucose molecules will be split into half to make 2 3-carbon chain sugars, and then they can enter the cell respiration cycle. So we start, our body will ingest something that's very large, break it down from the form of a polysaccharide to a disaccharide, and then down to monosaccharide. So another example of a polysaccharide that's found in organism tissue is that of cellulose. This is primarily specific to plants and algae cells, and this particular structure enables support for the plant and algae cells. The cellulose is something that we typically have a hard time breaking down, but there are some organisms that have the necessary enzymes in their gut, or release the necessary enzymes like protozoans, fungi, and bacteria that can degrade that cellulose and convert it to a smaller form to be used to construct energy for the cells. Okay, so now I feel like we've kind of exhausted our unit on carbohydrates with monosaccharides, disaccharides, and polysaccharides. So let's move on to another type of biomolecule, which is that of lipids. These lipids are very important to the body. They are not soluble in water, which means if you place them in water, they're going to be hydrophobic. But they're very essential to living because they can provide insulation, they can provide a location for storage in the body, and they assist in the digestion of certain things, making the amount of energy that your body needs to digest something a little bit easier. And it also keeps us warm, if I didn't say that already, through its buildup. But lipids are classified into several different categories. They can be a wax, which helps to retain or repel water. They can consist as a phospholipid, which basically means they have a standard lipid structure, but they're tied to a phosphorus chemical molecule. And you'll see phospholipids present primarily in the phospholipid bilayer that surrounds a cell. There's also glycolipids, which have a carbohydrate affixed to them, and then prostaglandins and leukotrienes, which are hormones that assist in growth and development. And then fats and oils are probably the most common that we're familiar with, as they assist in digestion purposes as well as storage of energy. And then steroids, which can enhance and also affect growth and development. So there's a variety of different categories that exist as lipids. Okay, so let's kind of look at the different structures of some of the lipids that we just talked about. Wax type of lipid, you essentially just have a fatty acid, which is basically a carbohydrate chain that has a carboxyl group at the end of it. And a carboxyl group is one carbon double bound to an oxygen that is bound to an OH group, or essentially a carbon bound to two oxygens and then bound to a hydrogen. But the structure reflects a double bond typically between the carbon and the free oxygen. And then at the end of that you have a long chain of alcohols that are associated or that are bound to the fatty acid. Triglycerides we're probably the most familiar with. This was, I think, one of the structures I showed you earlier. We're talking about unsaturated and saturated carbohydrates. You have a fatty acid. You basically have three fatty acids that are joined together via a glycerol stalk. And the glycerol stalk would be something simple like a linear structure of a carbohydrate chain. So you could have a 4-carbohydrate chain here, or a 3-carbohydrate chain, and then carbon 1 would be bound to the fatty acid, carbon 2 is bound to the second fatty acid, and carbon 3 is bound to the third fatty acid. But all these carbons are bound together, and so that makes what we refer to as the triglyceride. Okay, and so this is kind of the standard composition of what you would see with oils, such as like cooking oil. Phospholipids, you have, again, like I said, this is kind of, you would see this primarily in the phospholipid bilayer that surrounds a cell. But the structure here is similar to that of the triglycerides, but what has been replaced is the number of fatty acids. So on carbon 3 here, instead of having a fatty acid tail attached, you have phosphorus of the phosphate group, PO4, that's bound to an alcohol. And same thing with this sphingolipid, with the exception that you're missing the second fatty acid and third fatty acid. They've been replaced by this PO4 and then bound to chlorine. And then we've got glycoproteins, which are bound to the sphingosine, which is going to be the protein portion of this, and then you've got glucose and galactose, and then your fatty acid that'll also be expressed. And then we have a steroid molecule, which of all of these looks to be the most stable, primarily because it's written in or drawn in the molecular form. So these are typically just the general structures of some of the key lipids that are used by systems. So let's look at, again, kind of more in-depth structure of the lipids that are known as fatty acids. This, again, is a large carbon chain. There's probably about 17 carbons fitted on this chain, and it's bound to this carboxyl acid group that I was talking about earlier, where you have a carbon double bound to oxygen and hydrogen.